I know it's been a while, but welcome back to Don't Be Weak Science. I'm your host, Leader Han, and today we will be doing the Osmosis lab with dialysis tubing. Okay, so what you want to do is make sure you have three 250 milliliter beakers, and you want to label it like serum A, serum B, serum C. Okay. Next thing you want to do is you want to get the serum A sample, which I'll probably make for you guys, put it in like a 600 or 1,000 milliliter beaker. And what you want to do is you want to just pour it to about roughly the 200 milliliter mark. So like that. I mean, if you get a little bit above the 200 milliliter mark, that's fine. So A goes into A, B goes into your serum B. Okay. And then C goes into your serum C beaker. That. Okay. And you're going to return, the serum A, serum B, serum C beakers back to the center table so everyone can use them. Okay. Next thing you want to do is you can have your partner do this while one person's labeling and filling the beakers up. You want to cut about 10 centimeters of dialysis tubing. So you get a ruler, measure it like that. So 10 centimeters. Get a little bit more, a little bit less. That's okay. So cut it. Okay. And you'll need to cut out 3 of these. Okay, because you'll be making 3 dialysis tubes today. So you want to put this in water for about 3 minutes. Okay. Once they're done, this is one I already put in a couple minutes ago. What you want to do is you want to open up the dialysis tubing, and they're kind of tricky to open. But if you know the trick to it, then you should be able to do it pretty easily. So what you want to do is you want to put it between your thumb and your index finger, and just kind of rub it together, rub it away from each other like this. Okay. And it takes a little bit of time. Okay, there we go. So now we get an opening like that. And what you want to do is you want to get something like a pencil or like say a Sharpie, like that. No eraser in, okay? So probably Sharpie's better. And you want to kind of enlarge the hole like this. Okay. That way you keep it open. And you want to cut out a piece of string, roughly about this length, maybe about 10 centimeters or so. And you want to tie one end of the dialysis tubing. So what I do is I twist it. Okay, a couple twists like that. And then I tie the end. Kind of like a sausage, like I'm tying a sausage in. Okay. If you use any knot, I just use the shoelace knot. Make things easy. Make sure it's nice and tight, okay, but not too tight where you like actually rip the bag. I'm going to do another knot. Okay, just like that. Okay, and then you cut off the other end so that the string isn't too long. So like that. Okay, so you have one end tied like this, and the other end of the dialysis tube open. Okay, so what you're going to do next is you're going to get saline solution in a beaker. Okay, it should already be provided for you at your tables. Saline solution is going to represent basically the liquid portion inside our cells. Okay, the dialysis tubing bag is going to represent the actual cell membrane, and the cell membrane, they're semi-permeable, and just like a cell membrane, this bag is also semi-permeable. So you're going to attach the pipette on, and you want to get 10 milliliters of saline solution. Okay. Then you're going to put it inside the dialysis tubing bag. Like that. Okay. And then you want to close it. And then twist. Okay. You need another piece of string. And then you want to tie it. One more time. Make it nice and secure. Cut the end off. And what you want to do is, now you want to weigh this. So this would be your initial mass of one of your dialysis tubes. This actually represents a cell. Okay, so again, the dialysis tube is a cell membrane, which is semi-permeable, and then the saline solution is the contents of the cell. So you weigh it, you find a weigh boat, and then a balance, you weigh it, so let's say it's like 5 grams, okay, write down on your data table, and if this is, you need to make 3 of these. So let's say this is your cell that you're going to put in serum A, okay. Now, you won't know what serum A is, and then you'll have to determine that based on what happens to your cell. So is serum A, is this a hypotonic solution, meaning there's more solutes in the beaker, the fluid here outside the cell, or is it a hypertonic solution where there's more, I should say hypotonic solution where there's less solutes outside, or maybe it's an isotonic solution. Okay, the way that you're going to know that is this cell going to gain mass? Is water going to move inside, or is the water from the cell going to move outside into the beaker? And is this going to lose weight, or is it maybe it's going to be the same? So you're going to place the cell into the serum A, and you're going to leave it there for about 20 minutes. And you're going to do the same thing with a cell in serum B and then serum C. After about 20 minutes, you're going to pull it out. You want to dry it with a paper towel, so like dab dry it, but make sure you don't like puncture the bag. And then you're going to weigh it, and then you can determine the mass. So let's say that this was originally 5 grams, and then now it's 7 grams. Then what that means is water moved from outside of the cell to inside the cell. So under the rules of osmosis, that will happen if there are more solutes inside this cell and less a lower water concentration. So water will move from an area of low solute concentration to areas high solute concentration, and water will move from an area of high water concentration to a low area of low concentration. So that's what this cell would be, okay, compared to the serum A solution. So that would make this a hypotonic solution compared to the inside of the cell. Okay. After you're done with all 3 you recorded, then you want to clean up. So what you want to do is for serum A, serum B, serum A, B, C, you want to dump those into the sink, rinse them out, and then with your cell samples, you could, after you weigh them, you can toss them in the trash. Okay. And then that's it. Thank you.